Hello, everyone. My name is Anya Waite, and I'm co-chair of the SCORE Working Group 154, Integration of Plankton-Observing Sensor Systems to Existing Global Sampling Programs. And my co-chair is Emmanuel Boss at the University of Maine. We'd like to acknowledge the full members and associate members that did so much work in bringing this project together. Emmanuel and I were the co-chairs, but Sylvia, Elana, Marcella, Katya, Heidi, Sandy, Yulia, and Hidekatsu were all very, very helpful and important in getting the work done. Our associate members, Sonia, Jürgen, Hervé, Gerald, Johannes, Frank, Anthony, Bernadette, and Rick, were also very helpful. They came from many places across the globe, including US, Canada, Spain, Israel, Chile, South Africa, France, Japan, and Germany and Australia. So we had a very rich and interesting conversation. Our members included investigators from the United States, Canada, Spain, Israel, Chile, South Africa, France, Japan, Norway, and Germany. And we would like to extend a heartfelt thanks to everyone who contributed so much to this important project. The SCORE Working Group 154 was founded to identify the technologies and sampling protocols best suited to incorporate plankton measurements into broad global ocean observing platforms, and those include GoShip and Ocean Sites as a first case. So what did we do during these discussions? We looked first at essential ocean variables and essential climate variables and identified that microbial activity, phytoplankton abundance, zooplankton abundance, fish abundance, and suspended particles were the key variables that we wanted to focus on. And you can see in the brackets here, we then identified target technologies that might be easy to incorporate on board into some of the Ocean Sites and GoShip protocols. So why now? At a technological level, the technologies to measure biological variables are becoming mature, and many are very proven. The sensors are robust, they require minimal attention at sea, and the infrastructure for data curation and the protocols for data curation exist. Our working group started in September 2017. The first things we did were to source technology champions across the oceanographic community to review biooptics, flow cytometry, genetics, bioacoustics, imaging, and high-performance liquid chromatography technologies and techniques for potential integration into GoShip and Ocean Sites. Our kickoff meeting in February 18 was at Ocean Science at Portland, Oregon, and the discussions at this meeting resulted in a decision to focus entirely on commercial technologies so that whatever we recommended could be taken up immediately onto these platforms. So we didn't want things that are protocols on benchtops, even though there are many exciting protocols on benchtops that may supersede the recommendations that we make here. We also drafted an EOS article and an article in the L&O Bulletin, simply to tell the community what we were doing and to ask for their engagement and participation. In November 18, we had a second group meeting of the subgroup leads in Villefranche-sur-Mer in France, and we emerged from that with a first draft of the GoShip report and also contributed to a paper by Lombard et al. On globally consistent quantitative observations of planktonic ecosystems. In 2019, we worked on the editing and finalization of the draft GoShip report and produced that for GoShip and presented it to GoShip. We also went to eventually the full group meeting at OceanOBS in Honolulu. And by then we had also made a contribution to the Scientific Committee on Oceanic Research best practices system. And so our GoShip program recommendations are now freely available online, and we had several hundred hits within the first couple of weeks, so we were pleased with that. As we move into 2020, of course, things have been slowed down a little bit. We were originally going to be having an in-person meeting in September 2020, but this will now be an online group meeting, and we will finalize the draft Ocean Sites report by this autumn. One of the motivations was to make sure to include plankton essential ocean variables and essential climate variables into observing systems like GOSHIP. Plankton in GOSHIP are a critical component of the global ocean observing system. In the climate system, plankton fix carbon, transfer carbon from the surface to the deep ocean. They produce aerosols and seed clouds. Perhaps most importantly, plankton provide ecosystem health and ecosystem services. They provide biomass, diversity, life, and are the foundation of the global food web. As we move forward, particularly in this century, we need to start to inform policy with our observations, to inform the public on fisheries indicators, water quality, and progress towards the UN Sustainable Development Goals. We then reviewed all these water analysis technologies and sensors that have been developed to study plankton and looked at how they might mesh together to give a full picture of the planktonic ecosystem, from very small virus-sized particles on the left. If you look at the right-hand diagram here, if you start on the left with viruses and go right up to large medusae, you can see that we have a number of flow cams, cytobuoys, the list, the Coulter counter, and the underwater vision profiler, all of which observe at different scales across that spectrum. And on the left, you can see that when you use a range of instrumentation, you can actually get a clear picture of the particle size distribution and the size distribution of biomass generally across the whole marine ecosystem. So we then worked on these 6 broad categories: genetics, imaging, flow cytometry, pigment analysis, bioacoustics, and biooptics. We worked with 2 water sources, water samples that have to be analyzed onshore, and automated sensors. And then we had 3 collection methods. Of course, the hull-mounted things, for example, like your ADCP, which is acoustic sensors mounted on the ship hull, a CTD rosette, which is deployed usually off the side of a ship, and flow-through systems that are shipboard that can then be instrumented. We looked at how biological measurements could be done concurrently and on similar scales to physical and chemical characteristics. Our considerations were along a number of axes of discussion. First of all, we considered in the blue how quantitative a particular method was. Is there no quantification? Were there simply counts? Was it actually a biomass proxy? And could you actually move forward in having trait quantification along that blue axis? Then on the yellow, was this a very focused technology that worked only on one selected type, or could it be used across a full spectrum of plankton types and sizes? In the red axis, we looked at, could we see any distinctions between functional groups or the biological function of the groups? Or could we get full morphological taxonomy up to species level? Or were there no distinctions at all? And then in the green, the practical considerations: was this costly? How much expertise is needed to run the instrumentation? And how intercomparable are the technologies? And of course, the best along the green axis have highly scalable potential. So here's a very brief overview of some of the very long discussions we had. Bulk measurements and measurements of biomass have good scalability and are quantitative, but really give us very low taxonomic resolution and don't tell us much about a broad spectrum of organisms. When we look at pigments, you can see that there's a modest amount of taxonomic resolution. They're very scalable and quantitative, but again, they don't give us much biodiversity information. The quantitative imaging, on the other hand, gives us lots of information about taxa, also is quantitative, and it's scalable. When we look at genomic tools, the DNA sequencing, and so on. The taxonomic resolution is absolutely enormous, but this quantitative nature of this is still being developed, and it's very difficult to say in some cases whether or not the DNA is telling us exactly what we want to know about organisms or whether it's just telling us about DNA. On the other hand, it does cover an enormous range of taxa, from the tiniest viruses to the largest organisms. Bioacoustics has the advantage that it has very, very scalable and quantitative. However, of course, the taxonomic resolution is rather limited. And finally, biooptics, again, very scalable and quantitative, but has some of the same challenges that acoustics do. So these are some of the considerations that we incorporated into our final report to GOSHIP. So overall, our recommendations for a global plankton observing system are as follows. Methods should be standardized. This can be done, of course, through our global best practice system, which is currently under development and also supported by the IOC. Plankton samples associated with environmental variables need to be acquired simultaneously as physics and chemistry measurements. Sampling should include a complete spectrum of plankton size and function where possible. Of course, methods should be intercalibrated and protocols drafted, ideally through the best practice system. And finally, physical plankton samples need to be archived in the long term. And I'll just focus in on this for 1 minute because it seems like an extremely important aspect of what we did. When we ask the question, why archive samples? You can see, for example, that when you look at the Terra oceans in this picture on the right-hand side, you can see that the plankton here are changing the scale of their shell building, and that is likely because of ocean acidification. So we can see between the 1870s and the 2009-2013 sampling of Terra oceans, you can see a significant reduction in shell density and thickness. Now, this is an incredibly important thing to start to observe long-term as we try to understand the evolution of planktonic systems going into the future under climate change. So I'll just finish with our key publications included the white paper on recommendations for plankton measurements on the GOSHIP program with relevance to other seagoing expeditions, and this was primarily our SCORE working group report. We also did a community news article in the Limnology and Oceanography Bulletin, Beyond Chlorophyllescence, the time is right to expand biological measurements in ocean observing programs. Our capacity building activities, including publishing findings through ocean best practice, were somewhat limited in comparison to what we had originally hoped to do. However, we did do significant work within the US in particular to have teams proposing biological members for GOSHIP. In the US with OCB, we shepherded a team of early career scientists that submitted a proposal to the NSF. This was, in the first instance, declined, but they will resubmit. Individual members of our group work on initiatives, for example, a workshop on inline measurements that will take place at the University of Maine in 2022, and best practice documents which have been published under the auspices of the IOC. We'd like to thank the entire SCORE working group. The 154 team was great fun. We really enjoyed our sessions together, and I think we did make progress in our key areas. We'd like to thank SCORE for the funding. We'd also like to thank Patricia Miloslavich, our guardian angel, who followed us from proposal writing to reporting. We'd like to thank Ed Urban, who believed in our ideas and supported us throughout, and also to the Laboratoire Océanologique Villefranche-sur-Mer, who hosted one of our key sessions.